Bismillahirrahmanirrahim. Assalamu alaikum. This is Atifullah, student of BS7, Department of Microbiology, Kohart University of Science and Technology, Kohart. Today we will discuss a topic: transferable imipenem resistance in Pseudomonas aeruginosa. This topic has been taken from the paper of American Society for Microbiology. And this is the contents of my presentation, which include introduction, objectives, material and methods, results, discussions, at the end some references. Introductions. Imipenem is a broad-spectrum semi-synthetic beta-lactam antibiotic which works against the bacterial activity, including gram-positive, gram-negative, aerobic, and anaerobic bacteria. Imipenem bind with penicillin-binding proteins. Penicillin-binding proteins are the enzymes that involve assembling bacterial cell wall. When imipenem bind with penicillin-binding proteins, as a result, the bacterial cell wall cannot be synthesized, due to which the penicillin-binding proteins are inhibited by the imipenem. Imipenem is commonly used in combination with celastatin, which used against the urinary tract, respiratory tract, abdominal, blood, skin, bone, and joint infection. It is also available in triple drug product with celastatin and relebactam. It not work against the viral infection like flu and cold, etc. Recently, imipenem resistant clinical isolate of Pseudomonas aeruginosa have been found, and the resistance was due to decreased drug permeation with diminished production of outer membrane proteins. Enzymatic inactivation of imipenem has not been reported in Pseudomonas aeruginosa. We isolate a strain which produces an imipenem hydrolyzing beta-lactamase. The enzyme was a novel type of beta-lactamase mediated by a transferable plasmid. Objectives: identification of bacterial strain and isolation of plasmid. Material and methods. Bacterial strains and plasmid of Pseudomonas aeruginosa was a clinical isolate and collected in 1988 in Japan. Plasmid was isolated from Pseudomonas aeruginosa. Antimicrobial agents like cephalosporin, cephalotin, cefazolin, cefoperazone, etc. and some other antimicrobial agents like imipenem, ceftazidime, cephosium, and cefsolodine, etc. Susceptibility test can be done by an agar dilution method using sensitivity disk agar. Plasmid was transferred to Pseudomonas aeruginosa through conjugation and transformation. Determination of incompatibility was performed by the method of Sagai. Preparation of plasmid DNase and agarose gel electrophoresis was carried out by the method of Keto. Enzyme assays beta-lactamase activity was assessed spectrophotometrically with ultraviolet ray 265 spectrophotometer. Induction of beta-lactamase overnight culture was diluted 1:20 into fresh sensitivity test broth to a final volume of 100 ml. After 2 hours of incubation on a shaker at 37 degrees centigrade, cefazotine was added to a concentration of 25 or 100 gram per ml and incubation was continued for 2 hours. Results: we isolate imipenem-resistant Pseudomonas aeruginosa, which was also resistant to other anti-pseudomonal beta-lactams. Strain GN17203 produce imipenem hydrolyzing beta-lactamase. To determine the transferability of the imipenem resistance, conjugation was carried out with PaO1NF as the recipient, and transconjugates were selected on an agar plate containing ceftazidime and neuroproxazine. The second transfer were performed with a resultant transconjugate strain as the donor and PaO2142Rp as the recipient, and transconjugate were selected for resistance to ceftazidime and rifampicin. These resultants indicate that imipenem resistance was mediated by a transferable plasmid. The resistance to imipenem, meropenem, ceftazidime, cefsolodine, and carbenicillin was fully expressed in the transconjugates. The plasmid confirmed resistance to gentamicin and sulfonamides. The size of the plasmid was estimated as 31 MDA by agarose gel electrophoresis. Discussions. Imipenem-resistant clinical isolate of Pseudomonas aeruginosa have been found the lack of an outer membrane protein. The resistance was revealed to be due to decreased imipenem permeation with diminished production of the protein. We found an imipenem-resistant strain which produced a novel type beta-lactamase encoded by transferable plasmid TMS350. The effect of temperature on the activity of the TMS350 mediated enzyme was peculiar. The enzyme was most active at 60 degrees centigrade. Transferable R plasmid play an important role in rapid dissemination of resistance to antibiotics. This broad-spectrum enzyme will be one of the clinical problems in the therapy of Pseudomonas infection. And this is the some references of my presentation which has been taken from the research paper. If you have any question, please ask me in a comment box. Thank you for your kind attention.